Hello everyone, I'm Puspa Maurya and currently I'm studying MBBS. I am here to deliver my knowledge about bilysis synthesis, which mainly occur in the hepatocytes, and this occur by two pathway, that is first is classical pathway or neutral pathway, which accounts for 90% of the bilysis synthesis. That means it's the major pathway. And next is alternative or acidic pathway, which only accounts for 10% of bilysis synthesis. That means very few, a very insignificant amount of the bilysis or synthesis from the alternative or acidic pathway. So we know that the cholesterol are absorbed from our small intestine and then get circulated to the various tissues and organs of the systemic circulation. And we know when it reaches to liver, first it get accumulated in the cytosol of the hepatocytes. So from the hepatocytes, it needs to be transported to the mitochondrial matrix for its metabolism to bilysis. So what things in the hepatocytes that lead to the transport of the cholesterol from the cytosol to the mitochondrial matrix for its metabolism to bilysis. So we study about STARD1 shuttle, which is very important for the transport of cholesterol, which is the precursor of the bilysis. And this is transported from the cytosol of the hepatocyte to the mitochondrial matrix through this STARD1 shuttle. And this STARD1 shuttle is called as the steroidogenic acute regulatory protein. So if there is any dysregulation of the gene which encode for the STARD1 shuttle, or dysregulation in the activity of the STARD1 shuttle, then this may lead to the pathological conditions like Niemann-Pick disease type C. And next, it can cause hepatocellular carcinoma. Such pathological conditions may appear when there is dysregulation of this STARD1 shuttle. So next we see the steps involved in the bilysis synthesis. First step is initiation of the synthesis. As we previously discussed that cholesterol is the precursor of the bile acid, yes, and these bile acids are mainly synthesized from the cholesterol. Then what thing in the initiation stage lead to the two pathway, that is one classical pathway, next alternative pathway. What enzymes that lead to the two different pathway for the synthesis of the bile acids that we discuss in this initiation stage. Next is sterol ring modifications. We know cholesterol has a sterol rings, so its modification is very much important to get converted into the bile acids. So here we discuss about the sterol ring modification of the cholesterol for the synthesis of the bile acids. And next, third stage is side chain oxidation, and fourth stage is side chain cleavage. Here in the side chain cleavage, we see there is synthesis of primary bile acids, and this primary bile acids undergoes intrahepatic circulation and enters into the gut. And in the gut, the microbiota present in the gut lead to the conversion of this primary bile acid to the secondary bile acids. So these things we are going to cover in these things, and along with that certain disorders we will discuss further. And let's begin with the step 1, that is initiation state. Here as we know that the cholesterol metabolized to bile acid by two pathway, classical pathway and alternative pathway, and this um, classical pathway uh, undergoes 7 alpha hydroxylation. This is 7 alpha hydroxylation. This cytochrome P7A1 is also called as a 7 alpha hydroxylase that I have mentioned in this abbreviation, that cytochrome 27A1, that is called as the cholesterol 27 hydroxylase, whereas cytochrome 7A1 is called as cholesterol 7 hydroxylase. And this cytochrome uh, P7A1 enzyme lead to the 7 alpha hydroxylation of this cholesterol to get converted into this 7 alpha hydroxycholesterols. And next is uh, the cytochrome P27A1 enzyme, which is also called as um, cholesterol 27 hydroxylase, which lead to the hydroxylation of this cholesterol to 27 hydroxycholesterols. And this 27 hydroxycholesterol again undergoes hydroxylation by cytochrome P7B1 with the help of this enzyme. This again gets converted to 3 beta 7 alpha dihydroxy 5 cholestinoic acids. There is the formation of the acids. So that's why this alternative pathway is also called as acidic pathway. However, these both pathway lead to the synthesis of the bile acids, but here in classical pathway there is no intermediate where acid is present. So this classical pathway is also called as the neutral pathway, but alternative pathway is called as the acidic pathway. And this contribute for only 10% of the bile acid synthesis, and this contribute for 90% bile acid synthesis. And next, what we need to know that if there is any you know problem like non-alcoholic fatty liver disease, then there will be inhibition of this enzyme. In non-alcoholic fatty liver disease, the inhibition of these enzymes leads to accumulation of the cholesterols in the liver. So in the, even though person is not having alcohol, but there is inhibition of this enzyme cytochrome P7B1, then it may lead to the non-alcoholic fatty liver disease, as well as it also inhibits this enzyme. So in non-alcoholic fatty liver disease, this is the main enzyme that is inhibited. Next we see the sterol ring modification. In the sterol ring modification, we know here we, in the right side we discuss about classical pathway, and in the left side we will be discussing about the alternative pathway. We know that in the initiation step from the classical pathway there is the formation of 7 alpha hydroxycholesterol, and this 7 alpha hydroxycholesterol is again catalyzed by HSP3B7 enzyme, and from this catalyzation this is converted as 7 alpha hydroxy 4 cholesterol 3 1, and the same enzyme act on 3 beta 7 alpha dihydroxy 5 cholestinoic acid, which was already formed here in the alternative pathway. This again gets converted to the 7 alpha hydroxy 3 oxo 4 cholestinoic acid, the same enzymes, and this enzyme is 3 beta hydroxy steroid dehydrogenase type 7. So this enzyme is present in the endoplasmic reticulum of the hepatocyte, and this enzyme lead to the conversion of the intermediates of the uh, intermediate compound of the bile acids to 7 alpha hydroxy 4 cholesterol 3 1, and next is 7 hydroxy 3 oxo 4 cholesterolic acid. Next we can see that cytochrome P8B1, these enzymes lead to the 12 alpha hydroxylation of this 7 alpha hydroxy 4 cholesterol 3 1, and this leads to the 12 alpha hydroxylation. 12 alpha hydroxylation of 7 alpha hydroxy 4 cholesterol 3 1 uh, lead to formation of 7 alpha 12 alpha dihydroxy 4 cholesterol 3 1. And we can see that if the 12 alpha hydroxylation is not occurring, then this is converted to this 5 beta cholesterol 3 alpha 7 alpha diol, which is formed, which is also formed from the action of this AKR1C4 enzyme on the 7 alpha hydroxy 3 oxo 4 cholesterolic acid, which is the intermediate compound of the uh, alternative pathway. And this compound of the classical pathway undergoes to the formation of the intermediate compound of the alternative pathway if there is no 12 alpha hydroxylation. So this is the interconnection between the classical pathway and the alternative pathway. Next, what happens that this AKR1D1 or AKR1C4 enzyme act on this 12 alpha 7 alpha dihydroxy 4 cholesterol 3 1 and convert it to the 5 beta cholesterol 3 alpha and 7 alpha 12 alpha triol, and here is 7 alpha and 3 alpha 7 alpha diol. There is only diol because here is also no 12 alpha hydroxylation. 12 alpha hydroxylation is absent, and the intermediate of the classical pathway which is not undergoing the 12 alpha hydroxylation or again undergoes this formation of this. So there is no 12 alpha hydroxylation in these both. So only here is triol formation and here is diol formation, only at the third position and the seventh position, and here is 7 4 3 7 and 12th position, so lead to the formation of the triol. Otherwise it is same as 5 beta cholesterol 3 alpha 7 alpha and 12 alpha triol, and next is a 5 cholesterol 3 alpha diol. Here we can see. Now we go to the third stage, that is side chain oxidation, where 5 beta cholesterol 3 alpha 7 alpha 12 alpha triol, that is from the classical pathway, and this is 5 beta cholesterol 3 alpha 7 alpha diol, and this is acted by the catalyzed by the same enzyme. The same enzyme catalyze both and lead to the formation of here 3 alpha 7 alpha 12 alpha trihydroxy 5 beta cholesterolic acid, and here 3 alpha 7 alpha dihydroxy 5 beta cholesterolic acid synthesis. Next is side chain cleavage. Side chain cleavage of this compound, which was already formed here, uh, lead to the formation of the cholic acid, and here the side chain cleavage of 3 alpha 7 alpha dihydro 5 beta cholestinoic acid lead to the formation of the deoxycholic acid, and this is the primary bile acid that is synthesized in the liver. And this cholic acid and deoxycholic acid undergoes enterohepatic circulation, that undergoes the enterohepatic circulation, and when it enters into the GI tract, then the microbiota of the GI tract act upon these cholic acids as well as deoxycholic acid and convert them into the deoxycholic acid and lithocholic acid, and this is now called as secondary bile acid. This is how primary bile acids and the secondary bile acids are synthesized. And here we can see the summary of the bile acid synthesis, classical pathway and alternative pathway discussed. The initiation, the same cholesterol but catalyzed by the two different enzymes leads to the formation of the two different bile acid. So here we you can see the cytochrome P7A1 acting upon the cholesterol lead to the formation of the 7 alpha hydroxycholesterol, and the cytochrome P27A1 enzyme lead to the formation of 27 hydroxycholesterol, which again gets catalyzed by cytochrome 7B1 and 2 3 beta 7 alpha dihydroxy 5 cholestinoic acid, and this is the initiation step of the bile acid synthesis. Further, you can see that the same thing we discussed earlier, that the cytochrome P8B1 act upon this 7 alpha hydroxy 4 cholesterol 3 1 and lead to the formation of the um, 7 alpha 12 dihydroxy 4 cholesterol 3 1, and same this compound when not undergoing the 12 alpha hydroxylation, then this leads to the formation of the intermediate compounds of this alternative pathway, which is also found from this compound. Thus, the this intermediate, which when undergoes 12 alpha hydroxylation, then this leads to the formation of the cholic acid. But if there is the absence of the 12 alpha hydroxylation, then the same intermediate lead to the formation of the deoxycholic acid, and this much in the bile acid synthesis.